First, let me just say thank you for the opportunity to come here and speak to you today. Whoever chose the venue is brilliant. I know that you just had lunch, and the thought of just talking about insects or listing about insects might not be that appealing, but I'll try to make it as interesting as possible. So we don't have all the answers for these insects. They're very challenging, and as some of you might know, but we do have some topics that we've addressed. And so these are some frequently asked questions that come up. What species of Ambrosia beetles are problematic in ornamental nurseries and also landscapes? What are the symptoms of an infestation? What is their biology and life history? What trees do they attack? How do I monitor for them? And what management tactics are effective? So the black stem borer, Zalisandrus germanus, is the most abundant species that we have in Ohio, and it's also one of the most abundant species here in Kentucky. So this particular species was introduced, or it's native to Southeast Asia, and it was first reported from grapevines in New York, out on Long Island in 1932. And since that point in time, it's now found in 32 of 50 states. The males are flightless. You can see the male here. So it's the female that's making the decision on which trees to attack, and it's also the females that are important for identification purposes. Can you see okay for, okay. So the other species that people will encounter is the granulate ambrosia beetle, and that is also native to Southeast Asia, first reported from peach trees in South Carolina in 1974, and that is now found in 29 of 50 states. And Zalisandrus crassusculus is a little bit larger than Germanus. In terms of some general identification features, the head is kind of enclosed or covered almost in the pronotum, and they have this, it's like a, how do you describe it? There's like a ridge that runs along the end of the elytra, and there's also some very distinct stout hairs that occur there. And I'll address some more of the identification features as we go along. But in terms of symptoms of an infestation, so these are wood-boring insects. So the females tunnel into the stems of trees. Mostly the stem, sometimes branches, but mainly the stem. So it's a relatively small hole. It's only about 1 millimeter in diameter. And so the problem there is that the attacks are not always easily detected. And you can see here that those are tunnel entrances that have been created in dogwood and honey locust. So one of the characteristic features of these beetles are these soft, these sawdust toothpicks that will be seen sticking out from trees. Now, once rain occurs or if there's high winds, those toothpicks will disappear. So it's not a long-term type of identification feature, but it is something to be aware of. We also see defensive sap production, particularly in spring. But with honey locusts, we've seen honey locusts oozing sap in actually even late summer and fall. And so we've had growers notice this sap production and then realize that, wait a minute, there's a hole associated with that sap. And then, of course, when they cut into the trees, they see the tunneling by the ambrosia beetles. And this defensive sap production can be pretty copious and relatively large amounts that then, of course, is problematic from bacterial growth and other molds that can occur and make the trees unmarketable. We also see wilting and dieback. As you can see here, this is a magnolia, and this entire stem here is died back. And the basis for the dieback is really not well understood. As I'll discuss here in a minute, these beetles do have a symbiotic fungus that they carry, and that's really not considered to be highly pathogenic from a plant pathology perspective. So it's again, we don't really quite understand the basis for that. And it is also highly variable in the sense that sometimes trees will die back and other times they recover. So in terms of their biology and life history, so what are the females doing? Well, they tunnel into the stems and then create a series of galleries. And depending on the diameter of the stem, they may go in just superficially, or especially with this honey locust here, or with this magnolia, they've tunneled through basically the entire stem. And so that makes it challenging from a pest management perspective because now they are deep within the tree, and so that obviously complicates things in terms of chemical controls. So ambrosia beetles are one of the few examples of a fungus farming insect. And so what they do is they have a little pouch. If you were to slice the beetle longitudinally this way, you'd see a pouch within this region. And contained within that pouch are spores of their symbiotic fungus. So that's actually what they're feeding on. They're not feeding on the tree. So those sawdust toothpicks is just a result of the beetle chewing, and then she pushes that sawdust out and it forms this toothpick. But they're not feeding on the tree, they're feeding on the symbiotic fungus, and that's another reason why, as I'll mention later on, but that's another reason why systemic insecticides have not been found to be effective against these insects. One, because they're tunneling so deep into the tree, and two, because they are feeding on a symbiotic fungus. So in terms of the symbiotic fungus, one important concept that we think is actually a weak point in their biology is that the female will not start laying eggs within those galleries until the symbiotic fungus is established. So if that fungus does not begin growing within the galleries, as you can see here, she won't start laying eggs. So we think that that's a weak point in their biology that we can exploit. So we're trying to understand what factors are influencing the establishment of the fungus and then also the colonization success of the beetles. So in terms of their phenology, so they're overwintering as adults in trees. So they're not overwintering in the soil, they're not overwintering in other substrates, they're overwintering within the trees. And so those overwintering females will then emerge. In Ohio, it's generally late April to early May. In Kentucky, it's probably a little bit, well, it's certainly a little bit earlier than that, but I would think mid-April would probably be, might be the actual peak. But so we generally see 2 generations per year. And then those adults will then overwinter. In the fall, they will then overwinter again within the host tree. So for us in Ohio, the majority of attacks generally occur in the spring. We do get reports on occasion of growers observing attacks on trees In particular, it's always been honey locust, for whatever reason that is, that they've noticed attacks that have occurred in the fall, or what they thought occurred in the fall. But when we cut into those trees, we found that in fact there were males in those trees and larvae and pupae. But the fact that there were males there and the fact that males don't fly means that the only way that they got there was for the females to have produced them. And since the generation time is about 25 days from egg to adult, in other words, those attacks were first noticed in late August and September, but in fact they had been there for quite a while, probably this spring. So again, the majority of attacks that we see occur in spring. And that's also, as you'll see here in a minute, their peak flight activity. So this is mean number of beetles per trap that we've trapped in Ohio in 2015 in a woodlot. And I don't know if you can make out the dates, I apologize for that, but this is late April, May, June. And so again, we generally see a peak in flight activity that occurs in mid-May in Ohio, and that would just shift slightly within Kentucky and Tennessee to probably mid-April. And then we do see a second peak that occurs in midsummer. And these are corresponding to overwintering females that are then flying and looking for a host, and then the second generation here. We are currently catching beetles. I mean, you can't quite tell because this is a mean of 1,600 beetles that we caught within a week's time per trap. And so we are still catching beetles, and we do even have some experiments that are continuing right now. But again, in terms of what we've seen from nurseries, the most challenging times are in spring. And in terms of predicting the flight activity, some of you might be familiar with the concept of degree days, and basically that's using temperature to predict when an insect is going to be active. Well, the challenge with Ambrosia beetles is that they're already an adult, so they're already in the tree overwintering as an adult, so we can't really predict any development. But what we can do is predict their activity, and what we've found is that when there's 2 to 3 days being forecasted that are above 70 degrees, their flight activity dramatically increases. So in terms of timing applications of sprays, preventative applications, I know it's an incredibly busy time of year for nursery production, but when 2 to 3 days are being forecasted, that ideally you'll want to get your preventative applications on before those occur, because afterwards it could be too late. So yeah, in terms of first flight, March, I would think that Kentucky would fall in March to April, and then the more southern states is really February to March. So again, the beetles are overwintering in trees in woodlots, assuming there's no infestations within the nursery that have carried over. Basically, they're going to be leaving these trees within neighboring woodlots and entering the ornamental nurseries or the landscape and looking for a suitable host. So in terms of what trees do they attack? So this is where one of the reasons why they're so challenging from a management perspective, and that's because they have very wide host ranges. So the black stem borer, Zodosandrus germanus, can attack over 200 species of trees, and the granulate ambrosia beetle can attack over 120 species of trees. Now these are worldwide hosts, but the take-home message is that they have a very broad host range, that it's not a specific tree. And again, that's what makes it challenging. Now we do see that, somewhat, and this is regional too, but we do see that dogwoods, honey locusts, magnolias, maples, redbuds, and in Ohio, styrax. Those tend to be among the more commonly reported species that are attacked. Now, an important concept that will come up throughout the seminar is the role of host quality, the role of the actual tree, the physiological status of the tree. So these insects have traditionally been considered secondary pests of stressed trees. But there's reports of attacks on apparently healthy trees. And this concept of apparently healthy is a little misleading in the sense that just because a tree looks apparently healthy doesn't mean that it actually is. In other words, it can be emitting stress-related volatiles that the beetles are able to key in on. And I'll discuss that a little bit more detail here in a minute. But in terms of the distribution of attacks within a nursery, what we do find is that attacks are often clustered together. So all these black dots here represent trees, individual trees at a particular nursery within one of their plots. And the red circles represent individual trees that were attacked. The larger the red circle, the more attacks there were on that tree. So you can clearly see, based on a spatial analysis, what we were able to document was that, in fact, it's not a random distribution. So the beetles are specifically targeting certain trees. So even though they have a broad host range and they can attack over 200 species of trees, they're interested in attacking individual trees. And so the basis for that has been of interest for us in terms of a research perspective, because what is it about those particular trees that result in them being attractive to beetles and then subsequently attacked? Because again, if they, as a result of a broad host range, they're capable of attacking literally every species of tree within this plot, but they don't. So in terms of the host location behavior, so what these insects are actually doing to locate a suitable tree. Well, of course, visual cues play a role. The actual silhouette of a tree is important, but olfactory cues are even more important. And the olfactory cues, essentially the aroma of the tree, can really be divided into two categories. And the one is host-derived volatiles, so actually chemicals that the tree is emitting that the beetles use to say, okay, that's a tree I want to attack, particularly ethanol, which I'll discuss here in a second, and also beetle-derived volatiles, which are aggregation pheromones. So some insects will produce a pheromone to call in other beetles to then say, hey, help me overwhelm the host defenses of this tree. And so obviously that's important to understand for monitoring purposes, but also could explain why we see clusters of attacks or clusters of trees. In other words, that an aggregation pheromone is calling beetles into a tree, and they're then saying, okay, well, we're all going to attack that tree. Well, without getting into all the research, because basically it was all negative data in the sense that there's no evidence that these beetles are producing an aggregation pheromone. So in other words, what we see is that they're targeting certain trees for attack based on what volatiles, what attractants those trees are producing. And that's where ethanol comes in. And so it's actually really appropriate to have this meeting here today because ambrosia beetles have a very strong dependence on alcohol. And so basically in this particular experiment here, this is mean number of attacks per tree. And so we took an ethanol lure, one of these little lures here that emits ethanol, and hung it from trees, and then also had trees that we didn't hang ethanol from. And so as you can see here, on the trees that we hung the ethanol lure on, we had about an average of 15 attacks per tree, and no attacks on the trees that were unbaited. But as you can see, the attacks stopped at 15. So basically, once it hit 15, there were no longer any attacks. Well, that's where we actually purposefully removed the ethanol lure from that tree. So as soon as the ethanol was taken out of the system, that tree was no longer viewed as a vulnerable host. So we've actually figured out that instead of using a pressurized injection system for injecting insecticides, you can actually inject trees with ethanol. And when you do that, depending on the concentration, the more ethanol you inject, the more attacks you get on those specific trees. And here you can see the amount of sawdust piling up around those trees. So beetles are very attracted to trees that are emitting ethanol, and neighboring trees that were injected with water and not emitting ethanol were not attacked. So just to give you an idea of how efficient they are in finding trees that are emitting ethanol, we did an experiment where we set up a tree in the center of a plot that was injected with ethanol, and then neighboring trees were placed around it that were not injected with ethanol. So they were not, to our knowledge, stressed. They were not emitting ethanol. So then we looked at landing rates and also attacks on these trees. So we found that there were no attacks on any of the trees despite their proximity to the ones that were emitting ethanol. So if they were not emitting ethanol, no attacks on those trees. And beetles actually rarely landed on the trees that were not emitting ethanol. So essentially what they did was they left the woodlot and flew right towards the tree that was emitting ethanol. And those were the ones that were attacked. We had an average of 17 attacks on these trees in 2011 and 26 in 2012. So again, a very efficient ability to find trees that are emitting ethanol. So why ethanol? Well, ethanol is emitted in response to a variety of physiological stressors, and a number of these can occur within ornamental nurseries, within the landscape, especially if there's an extreme climatic event. And an important concept to be aware of is that the tree that's under physiological stress can begin emitting ethanol within 1 to 2 days of that stress being initiated. And so to a grower, to a researcher, that tree could look perfectly healthy, but yet it's actually emitting stress-related volatiles in response to that stress. And that is then signaling to the beetles, I'm a weakened host and suitable for colonization. So again, the concept of apparently healthy, yes, it may look apparently healthy to us, but what's actually occurring in terms of its interactions with beetles. So again, they can be asymptomatic or apparently healthy, but still emitting ethanol. So in terms of flood stress, when a tree is, when the roots are deprived of oxygen, the tree will switch from aerobic respiration to anaerobic respiration, which is similar to what they're doing here at the distillery. And so a byproduct of that is ethanol. And so as a result of a tree being flood stressed, it will then emit ethanol. So to give you some examples, some field examples, we had a grower that called and said that he had lost every one of his dogwoods in this particular plot that you can see here. And no neighboring species of trees were attacked. They certainly were on the species list capable of being attacked. So the grower concluded that he had lost all these trees to ambrosia beetles. So we looked and we examined 166 of these trees, and 99% of them exhibited dieback, as you can see here. But only 70% of these trees were actually attacked by ambrosia beetles. So in other words, there was already the ambrosia beetles were acting secondarily. That these trees were not killed by ambrosia beetles. There were other issues that were going on that attracted beetles to these trees. And so when we looked at some of these trees, in particular this one, no ambrosia beetle attacks, but exhibiting dieback. And you can see this chlorotic zone moving up from the roots. And the planting depth also appeared to be way too deep for dogwoods, as you can see here. So it was really a combination of that particular year. We also had an extreme, one of the wettest seasons on record in Ohio. And the grower felt that looking back, the grower felt that he really should not have planted dogwoods in that field because it was the most low-lying field in his nursery. The water just came in there and sat, and obviously that was a problem. So we've conducted, as a result of those observations, we've conducted actual experiments looking at our ability to use flood stress to induce attacks on trees. And in fact, it does. And so an important concept that we considered was that depending on, of course, there's ranges in variability of tolerance and intolerance to trees. So some species can be tolerant of flood stress and some species can be very intolerant. And that influences their ethanol production. I mean, there are some exceptions, but generally speaking, an intolerant tree is going to switch over to anaerobic respiration and then begin emitting ethanol. Whereas a tolerant tree would not be emitting as much ethanol. So we thought, okay, well, let's give beetles a choice among these trees and see what happens. So we flooded different species of trees that were either tolerant or intolerant, and then of course also had trees that were not flooded, and let beetles make a choice on which ones to attack. And so the slide's a little busy, I apologize for that, but basically these are a number of attacks here on the y-axis. And so what we found was that, what we found was that the trees that were characterized as flood intolerant, particularly the dogwoods and redbuds, sustained the majority of attacks. Whereas the moderately tolerant to intolerant trees were not attacked. And of course, the non-flooded trees did not have any attacks at all. So the take-home message here is that flood stress is definitely not accounting for all the instances that growers are dealing with ambrosia beetles and losing trees to ambrosia beetles. But there are certainly a number of examples that we've seen in a number of states, not just Ohio, but Virginia, North Carolina, where trees that are being overwatered or not draining properly, if it's a pot-and-pot system and those pots are not draining, that if those water levels are allowed to build up and it's an intolerant tree, then that's where you're going to have issues. And I should point out that we analyzed these trees for ethanol, and we did find that ethanol levels were significantly higher in the intolerant species versus the tolerant species, and no ethanol was detected in the non-flooded trees. So we know that these insects are opportunistic colonizers of stressed trees, and up until last year, we had never seen attacks on a neighboring tree that was not stressed. So in other words, these trees, these numbers under trees are the total number of attacks on those trees, okay? So this flooded tree, dogwood, had 173 attacks. One next to it that was not flooded had 0. And this one had 260 attacks on the flooded tree, 0 on the non-flooded tree. Well, you can see here that this tree had 338 attacks, and the non-flooded tree next to it had 1 attack. And then also there was 8 attacks on this non-flooded tree. And that was really surprising to us because we had done a number of experiments and had not seen this before in a number of locations. And so this pertains to the concept of spillover. Can beetles spill over from attacks on a stressed tree? Can they spill over onto a neighboring tree? So obviously from a management perspective, it's an important concept to be aware of. And so when we actually looked at the tunnels associated with these non-flooded trees, They were just superficial tunnels and absent of beetles. So yes, they tunneled in, but they abandoned that tree. So we then followed that up by caging beetles using what would be referred to as a no-choice test. So we took small cages and forced beetles to be exposed to either flooded or non-flooded trees. And we then measured the amount of sawdust that was collected within those chambers, and there were significantly more sawdust in the flooded tree versus the non-flooded. And we found eggs, larvae, pupae, and the symbiotic fungus were only found in the flooded trees and not the non-flooded trees. So the take-home message here is that these insects are very poor colonizers of healthy trees. But if it's a weakened tree, especially one that's associated with ethanol, then their colonization success increases dramatically. And what we're thinking now is that we have evidence that ethanol is actually promoting the symbiotic fungus, the growth and the establishment of the symbiotic fungus. Because again, she will not start laying eggs until that fungus is established in the tree. So when they tunnel into a healthy tree, perhaps they're unable to get presumably, they're unable to get the symbiotic fungus to establish, and they leave. So in terms of drought stress, this is an experiment that we did where we allowed beetles to make a choice among flood stress, drought stress, and then of course untreated. And these are the attacks on the flood stress tree and no attacks on any of the drought stress tree. Now, we have seen issues where drought stress trees have been attacked under experimental conditions, but very low levels. And that's probably because for growing the symbiotic fungus, a drought stress tree is probably not a very good location. So certainly flood stress is important. Drought stress, I would think that if a tree was drought stressed and then perhaps heavily watered or then subjected to overwatering, then there could be some issues there. But so just in terms of other stressors that we are beginning to realize are important, there was an article that was published in 2012 from Belgium that focused on the same species, Alexandrus germanus, that we're all challenged with here. And they found that frost injury was increasing the susceptibility of trees to Ambrosia beetles. And basically what they found was that large-scale outbreaks of attacks were preceded by extreme frost events. So we saw, if you remember back, there was a mild winter in 2011, 2012, and in Ohio, we had April actually being colder than March. So trees were about a month ahead of schedule and then subjected to April frosts, which is not unusual to have a frost in April, but the fact that the winter was so mild, those trees are a month ahead of schedule, we then had indications of frost injury that were then predisposing trees to attack. And we had a grower call about Kentucky yellowwood that was growing in his front yard, and it was a specimen tree, and he had never had any issues with it. He called and said, "It's my wife's favorite tree. You've got to come over to the house and take a look at it." And so we did, and you can see the staining from the Ambrosia beetles. And this was a very large diameter tree. And so we started talking with him, and he said, "Oh, you know what? This tree tried to leaf out twice." You can see here, and it was zapped twice. So that foliage was zapped, and then beetles started attacking it afterwards. So initially, his thoughts were, well, Ambrosia beetles just decided to start attacking this tree this year. Then it was, well, wait a minute, okay, the attacks were preceded by 2 frost events. And we've done some experiments where we took styrax, which we've seen from other nurseries that have been frost injured and then attacked. So we frost injured them under controlled conditions and gave beetles, put them out under field conditions and allowed beetles to attack them. And the frost injured trees were preferentially attacked, as you can see here. And we've also measured ethanol levels over time, and we pick up ethanol levels within 1 day of those trees being frost injured. And actually here you can see that 1 day after we put those trees out, beetles started attacking those frost injured trees. So again, that's where it's so challenging, because they're rapidly colonizing these trees. I mean, they really don't waste any time. So we've also seen some field observations of winter injuries. So the whole polar vortex, extreme winter temperatures that occurred in 2014 and 2015. And here, this particular maple, you can see has winter injury, some bark splitting here, and maybe some of you can make out that the attacks are circled here in blue. And these 3 trees here that have the ribbons on them, those were attacked. And so again, we saw instances of clustering of trees. So oftentimes if you find one tree, there's a good chance that there's another attacked tree close by. And the basis for that, we think, could be localized site conditions or localized microclimates that are resulting in issues with that area. We don't think it's a stressed tree next to a healthy tree and spill over onto those healthy trees. We think that these trees are physiologically stressed and then being attacked. And I should also point out that 2014, following this extreme winter injury, there were large number of reports from the tree fruit industry in New York and Virginia and Maryland about, and perhaps Kentucky, of Ambrosia beetle attacks. And they were considering it to be a new pest. And in fact, I mean, those beetles have been established in those areas for long amounts of time. And so it's in my opinion, it's probably more of a scenario of the rootstock or the cultivars that are being used currently perhaps are not as tolerant as one might think for the extreme winter conditions that occurred. And so that's why, as a result, you saw so many issues within the tree fruit industry. And here, I mean, not surprising, but here's there's some extensive bark splitting, and when we peeled it away, you could see some sawdust from the ambrosia beetles attacking those trees. So in terms of monitoring, well, as you can imagine, ethanol is the most attractive compound that we know of for these insects. There are some compounds that can enhance the attractiveness. Canopterin is one compound that some of you might be aware of. And we have seen instances where it does increase trap counts, but other times it has a negligible effect. So at this point, and it's not cheap, the canopterin. So really, I mean, just an ethanol lure that you can buy in these packs commercially is the way to go. You can make them using like a bottle with a wick, but there's 2 challenges to that. One is we had a really difficult time finding ethyl alcohol. I mean, I guess you could use cheap vodka, but we had a really difficult time finding ethyl alcohol, and you don't want to use isopropyl alcohol, rubbing alcohol. So that's the one issue. But if you can find ethyl alcohol, the other problem with making your own is the release rate. You'll probably find that if you make your own, you're going to have to refill them every 2 to 3 days. And so, whereas if you purchase them commercially, they have it's more of a a much more longer release rate. I mean, you can use these packets for a couple of months. So in terms of the traps, this is where you can actually save some money and make your own trap. So a lot of people will just take a bottle and turn it upside down and just cut some holes in the side, and then you can put soapy water in the bottom, and then you want to position that low to the ground, and you can then look for your insects. The key, of course, is to place the traps low to the ground, because these are low-flying insects. And that might be because, I think that's probably because ethanol is heavier than air, so it will actually settle down and be low to the ground. So that's probably why they're flying low to the ground. I should mention this is another trap. This is called a Lindgren trap that you can purchase commercially. It's very common for forestry applications for bark beetles, but and it can be used for ambrosia beetles, but it's very expensive, and I think that using a bottle trap is really, in this case, an opportunity to save some money. You will see people mentioning the use of antifreeze as a preservation technique or as a killing agent, and that's fine. I mean, we use it for research purposes, but you just want to make sure that if you're using this type of trap that you don't want to put antifreeze where it could then be easily consumed by a pet or wildlife. So in terms of monitoring flight activity, you can also soak a bolt in ethanol for 24 hours and put that out, as you can see here. Or you can use a flood-stressed tree. And what's nice about that is you can actually visually see the toothpicks, and that will help you educate yourself in terms of identifying what the beetles look like, because you can then cut into it. And even with the naked eye, I mean, you'll be able to see. I mean, you're going to get some bycatch in these ethanol-baited traps, but during peak flight, you'll be able to very quickly see, oh, these are clearly dominated by ambrosia beetles. So in terms of management tactics, and this is true for integrated pest management in general. The foundation of an integrated pest management program, the #1 step is to maintain tree health. That's the first step in an integrated pest management program. And that's especially important with ambrosia beetles. Now, I know it's very easy for me to say, well, the trees are being stressed and that's the problem. But the hard part then becomes understanding what factors predispose those trees to attack, and then how can those be addressed. And that's where it's really important to get feedback from growers and to interact with growers and be familiar with what's happening in terms of the nursery, because I mean, you're the ones that are out there and you're the ones that are going to be seeing these things firsthand, and so it's important to understand those concepts. And so but again, we do consistently see that flooding, frost injury, and winter injury are problematic. So in terms of preventative chemical controls, I will say that none of the insecticides that we've tested are 100% effective, but permethrin and bifenthrin-based insecticides are generally the most effective products that we see. We've not seen systemics, we've not found systemics to be effective, unfortunately. And the applications should be applied as much as possible to the trunks of the tree, or the main stem, because as you can see here, 90% or more of the attacks occur on the trunk. Now, it's ideal to get the treatments on before peak flight. Now, this is just to give you an idea of the importance of maintaining tree health. For instance, these are attacks on a flooded tree. These are attacks on a flooded tree that was treated with permethrin. And then here's your non-flooded trees that were either untreated or treated with permethrin. So yes, we can reduce attacks on those trees, but again, maintaining tree health is the key, because insecticides reduce but do not always prevent attacks from occurring on stressed trees. But we are seeing that the colonization success is being somewhat reduced by insecticide applications. So galleries with fungal growth were significantly reduced in flooded trees that were treated with permethrin. So that's encouraging. And galleries with eggs were also, actually we didn't find any galleries with eggs in flooded trees that were treated with permethrin. So again, in addition to just simply reducing the number of attacks, we're also finding that preventative applications are in fact reducing the colonization success. Efficacy of fungicides. This has come up, and we haven't, I mean, you'll see in extension-based literature that fungicides don't work, and even after the trees have been attacked. But, and that's probably true to a certain extent, but there's really not a lot of, actually I haven't seen really any evidence in terms of actual testing of fungicides. But, so we're starting to look into that. But we did find that preventative applications of these two fungicides did not reduce attacks, not surprising, compared to a flooded tree that was not treated. But we did see that galleries with fungal growth were significantly reduced with a pre-treatment with a fungicide. So again, it might be cost prohibitive, but it does seem like there is some encouraging evidence in terms of disrupting the colonization success. And again, galleries with eggs, we didn't find any galleries with eggs in trees that were flooded and treated with the fungicide. So we're also looking into rescue treatments. So in other words, the thinking for a long time has been, okay, the trees are attacked, cut them down and put them in the burn pile and burn them immediately. Or leave them in place for a couple weeks because they'll still be attractive, they'll attract beetles, then cut them down and get rid of them. Well, we're kind of changing our thinking about that because trees can recover from ambrosia beetle attacks. It's not a death sentence. And so if you do have trees that have been attacked, if they're not exhibiting wilting or dieback, and I'd say if the attacks are maybe under, maybe a dozen or even 2 dozen, you can try to leave those trees in place and see if they recover and heal over. And we have seen examples of that. In addition, we've also begun looking at rescue treatments. In other words, after a tree has been attacked, can we apply a product and try to get some disruption of that colony? And so we flood stressed trees, allowed beetles to attack them, and then treated, after 2 weeks, we then treated with baseline, which is bifenthrin, and agrifos. And we did find that there were significantly more dead beetles and associated with the trees that were treated with a combination of baseline and agrifos. So again, the current thinking is that even though the beetles are in the trees, we might still be able to reduce the severity of that colony rather than just saying, well, cull the trees. One of the other concepts that comes up a lot is a push-pull scenario. Can we use repellents to push beetles away from vulnerable trees and then use attractants like ethanol beta traps to then intercept them? And so the idea, for instance, you could put your ethanol beta traps along the perimeter and try to intercept those beetles and then integrate repellents within the nursery. So we did an experiment in Ohio, Virginia, and Mississippi last year. And so basically we had flooded trees that either had a verbenone, which is a type of anti-irrigation repellent, either had the repellent or not, and then either had a perimeter of an ethanol lure or not. And unfortunately, as you can see here, this is the number of attacks on these trees. There was no significant differences in Ohio and Virginia. In other words, the perimeter traps did not prevent those trees from being attacked, and the verbenone repellent didn't reduce any attacks either. So we were kind of discouraged by this. Our colleague in Mississippi, he found that having the perimeter of ethanol baited traps did reduce attacks on those trees, so it was beneficial, but he also had a very, very low level of attacks. I mean, like maybe an average of like 2 to 3 attacks per tree, whereas we had much higher levels in Ohio and Virginia. So maybe under low levels of pressure it might work, but it might also be too that we have to try a higher release rate and make these traps even more attractive, but we're still looking into it. So in terms of management plan, just to summarize, maintain tree health, consider physiological stressors, monitor spring flight activity with ethanol baited traps or ethanol soak bolts or both. Pre-treatment with permethrin prior to spring flight activity. I mean, as far as applying throughout the season in the fall or midsummer, I mean, I don't necessarily think it's probably necessary. I mean, it's hard to say, but but we generally see that the majority of our problems are occurring in spring. And in Ohio, I don't think any growers that I know of are really specifically doing any applications in late summer or fall for Ambrosia beetles. But they have in the past, and I certainly understand if a grower has had issues and is concerned about losing a crop. So it's kind of a gray area right now. But and yes, if a tree is very heavily attacked, you can leave it in place and attract more beetles specifically to that tree and then discard that tree. But again, it's not a death sentence for the tree, and in some cases they can definitely recover. And so with that, I have a number of collaborators that are listed here in terms of funding. HRI and the Floriculture Nursery Research Initiative have been very supportive. And so hopefully I didn't go too far over. And my cell and email are on here, so please, if you think I can help, give me a call or send an email, and I'd be happy to talk. Thanks, appreciate your time.